Muscle contraction!
This is the muscle. Muscle is composed of fascicles. Each fascicle is a group of myofibers. Each individual myofiber is composed of myofibrils, or lesser units.
The sarcolemma is the cell membrane that covers the muscle fiber.
The T-tubules, or transverse tubules, are where the sarcolemma dips into the myofiber. Inside the myofibers, there are mitochondria that produce ATP.
This is the sarcoplasmic reticulum. It is composed of T-tubules and calciquestrin.
This is the sarcomere. The sarcomere is the basic functioning unit of a striated muscle. It is composed of thick and thin filaments that extend from Z-disc to Z-disc.
The darker middle part of the sarcomere is the A-band, which extends the entire length of the thick filaments. Toward each end of the A-band is the zone of overlap where the thick and thin filaments lie side by side. A narrow H-zone in the center of each A-band contains thick but no thin filaments. The I-band is a lighter, less dense area that contains the rest of the thin filaments but no thick filaments.
This is a thin filament and a thick filament. On the thick filament are myosin heads. On the thin filament, there is a tropomyosin covering the active sites on the actin. On the tropomyosin is the troponin.
Muscle contraction begins with an action potential that runs down the axon. Depolarization opens the calcium channels and the calcium ions pour in. The calcium causes the synaptic vesicles to move toward the cell membrane. The synaptic vesicles fuse with the membrane, releasing acetylcholine into the synaptic knob by exocytosis. The acetylcholine attaches to its receptors, which open the channels. When the protein channels open, it allows sodium to flow in. The acetylcholine is then broken down by acetylcholine esterase into acetic acid and choline. Choline goes back into the synaptic knob, while acetic acid and sodium enter the muscle cells.
When calcium is released, the troponin reacts, pulling the tropomyosin off of the active sites and exposing them. When ATP reacts with the myosin heads, a phosphate group breaks off, releasing energy. The myosin heads then go into a high energy state, reacting with the active sites that have recently been exposed. The reaction of the myosin heads with the active sites causes the muscle to contract.
After a muscle contraction, calcium is drawn back into the reticulum where it is stored until the next muscle contraction.
Myosin heads pivot to the low energy state, releasing ADP. The thin filaments move over the thick filaments. And the muscle is relaxed.